Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thank you very much, Mike, and thanks to the organizers for inviting me, and it's a real pleasure to be here. I think it's actually a great opportunity to connect back some of the things that are going on in genomic sequencing, to the community here. As Mike says, I was sort of somewhat involved in the machine learning community about 25 years ago, and then shifted off as sequencing the human genome started. So, we're in a special time for biology where over the last 20 years or so, we've moved from having extremely sparse information about the genome sequences of organisms to nearly complete information. And I didn't find myself a pointer, but I guess there is one somewhere. But in the 90s, as you can see, over a period of just 5 years, there was a 3 orders of magnitude increase in the size of genomes that were sequenced. And that rate of increase of the ability to obtain DNA sequence didn't stop then. It didn't stop at the end of the Human Genome Project. Between 2000 and 2010, costs dropped by another 5 orders of magnitude, much faster than Moore's law. And that's pretty daunting. And it creates petabytes of data a year now, and it's seriously demanding for computational, for data analysis and interpretation. So, initially we started by thinking we'd get one sequence of each species, like the human genome sequence. But of course, there isn't one human. There are billions of humans, and we all have different genome sequences. And a lot of what is interesting is what is different between them, and how that relates to the differences between the individuals. So we can now study genetic variation, which has been an important field for biology for the last century, but it's always been done in the dark up until the last 10 years or so. We can now do it directly in the light. We can sequence and see what we're working with. So, as you can see in terms of costs, the first human genome cost about $1 billion. By 2009, we sequenced 200 genomes at about $50,000 ago. That dropped last year, or by this year, we're into the tens of thousands of human genome sequences. Costs are below $5,000. It depends, exactly what deal you get from whoever. And I'm pretty confident that costs will continue to drop. There are new companies, new ideas, technological ideas for collecting data. We'll get human genome sequences below $1,000, and there'll be millions of human genomes for clinical usage, for research, and for personal interest as well. So, we need to somehow make sense of all this information. And in doing that, it's probably, it's not a surprise to you. I think we need to make use of the structure in the data, both for representing the information effectively and for inference on it. And, it's not quite as scary as it might seem, because the more people you sequence, the more they look like each other. We're all related. We're all related by evolution. And effectively, at each place in the genome, there's a tree that relates us. And so there's a common ancestor some time ago, about 500,000 or 1 million years ago on average. It's quite high variance. And we're all descended. All the different versions of the genome, the several thousand or the 1,000 or so that are in this audience, are derived from that. And the differences that we observed are caused by mutations that happened since that common ancestor. And so they're shared typically by some subset of the people. And that sharing means there's a allele frequency. There's a frequency that the variant has in the population. So you can think of genetic variation as composed of binary events, which were mutation events, and there are those people who have the ancestral form and those who have the derived form. Okay, so we get different events here at different times, which lead to different allele frequencies. And for those who are doing this sort of standard association genetic studies for disease, some mutations might cause disease or have some other property that is visible, has a visible effect. And you can just count up. You can take people marked in D here who have the disease and those who don't, and you can ask, is there a statistical association between those with disease and the genetic variant? And there's a huge power to that, because although normally correlations or associations are sort of, you have to be a bit suspect, you don't know about causality. With genetics, you essentially know about causality. You know that it's not true that having the disease changes your genetic state, or caused this mutation to happen 100,000 years ago. So you're pretty confident that there's a causal direction from the genotype to the phenotype, to the disease. So that would make everything very easy. We know how to do lots of things on trees. Trees are nice things. There's lots of algorithms on trees. Unfortunately, life isn't so simple. The tree, there isn't just one tree. The tree relating as changes as you move along the genome. So at each position or locus, there's a tree. But as you move along, there could be ancestral recombinations, which I'll describe what that is in a moment, which will change the tree. And so you get this complicated graph. And the one way to think about it is, we think about a tree going back to a common ancestor in time at one place. But you also have an inverse tree, your pedigree of all your parents, your ancestors. And those increase going backwards in time. And in some sense, the real structure sits in between those two. So let's consider a simple case of 4 individuals. And these are 3 variable positions. Here, the A is a C, and B, C, and D are T. Here we've got A and B are Gs, and C and D are As. And here A and C are As, and B and C and D are Cs. So we could represent them in binary. We could show a tree that explains the first position. There's a mutation that where A is the ancestral 0, B, C, and D are all 1. On that same tree, we could put the second mutation. C and D carry the mutation, so we can place it there, at that point in the tree. But there's no way we can place a third mutation on that tree to give rise to the pattern that we see for the third site. And in fact, there's no tree on which we can place 3 mutations. And that's very straightforward to show. So such patterns do exist, though, in real data. And what's going on is that the tree is different. And so what might have happened here is that C is no longer closely related to D, but is actually most closely related to A. So then everything works out fine. You can place the mutation here, and we understand what's going on. So and what's happened is that at this point, somewhere between this position, the left side to where these 2 mutations are, and the right side, the person C inherits their DNA from a different person in the past. And we know that you inherit your DNA from your grandparents, and you get a mixture of your grandmother's and your grandfather's DNA forming each of your chromosomes. So you get these events which splice, which cut at some point, switch over from grandmother to grandfather. And that's exactly what we're seeing here, and we can represent that in a graph that has bifurcations both going backwards in time and forwards in time. And this bifurcation is due to a recombination. It's a bringing together of pieces from 2 different individuals in the past into a modern single piece of DNA. And in general, you get this big structure, interesting structure called So that's the real structure. There is such a structure that relates all the DNA of everybody in the room. Unfortunately, inference on these things is hard. So that's what we want. That's the right thing in some sense. There's a natural generative model for them. It's the coalescent with recombination. Kingman introduced the coalescent, for those who know about this, in the 70s, 80s. Tavare and Griffiths introduced or looked at recombination, and there's been a lot of work and so on. They're good simulators. There's also an extremely good Markov approximation, which essentially runs left to right and says you have a tree at one position, and you can sort of do operations where you break a branch on the tree and glue it back somewhere else. We can think, since it's a tree, we can use a horticultural analogy and talk about pruning it and grafting it back, which is what you can do with real trees. So you can have this Markov process where the hidden state is a tree, and you observe mutations, you observe sequences on the leaves. Unfortunately, there are infinitely many ARGs, ancestral recombination graphs, for any particular data set, if you have a set of data. And sampling histories is is an unsolved problem, actually. There's MCMC strategies. They're extremely costly. It's a very hard space to search. And also, conditional sampling is hard. If you have a set of data, if you want to sample and choose another individual or place them into that structure, that's a non-trivial operation. So that's a sort of framework. We're going to want to come back to this in the future, because that's really what we're going to want to play with. There's one more thing that's important, which is that there are high demands on accuracy. So this is a section of the genome. It's about 1,000 bases long. You can't see very well. There's a few letters. Doesn't matter, really. These are actually Craig Venter and Jim Watson's sequences. This is an old slide. There's one place where they differ on this slide, and that's typical. There's 1 in 1,000 places. And they both differ because they share there's a variant that's shared. So if we want to have a 1% sort of error rate, and we have a 1 in 1,000 places overall, which are different, we need a 10 to the minus 5 per base pair sequencing error rate. But if we want to look at 1,000 people, we need a 10 to the minus 8 per base pair sequencing error rate. Raw sequencing accuracy is at best 10 to the minus 4. So we're going to have to use statistical methods and to integrate raw data, whatever we do, to get at our sequence. So let's just keep that in mind. So I'm going to tell you a little bit about the 1,000 Genomes Project, which is this major international consortium that, as sequencing costs dropped, led the way, essentially, in sequencing multiple people to characterize genetic variation. And to motivate what I'm going to go on to later, which is a new way to try and carry out inference on ARGs in an efficient fashion that hopefully might stimulate some ideas. So the 1,000 Genomes Consortium was put together in 2007 with a number of centers from across the world, the Broad Institute in Boston, Baylor College of Medicine, WashU in St. Louis, Sanger Institute in UK, and the BGI in China. Those are the main centers, but also some of the manufacturers of instruments and funders, of course. And we published pilot data on 190 samples in 2010. We sequenced 1,000 samples in 2012, and essentially complete now with completion next year. And the aim was to find essentially all the common variation, to sample enough individuals from across the world to find things that were shared by at least 1% of people. Try and be a bit more accurate and deeper in the coding sequences. To also discover other types of genetic variation, as well as single point mutations. You can delete or rearrange DNA. And to identify a reference set of human sequences from the individuals we sequence. And it's been a driver for the data. A lot of the methodology that's used in sequencing experiments now came out of this project. So we started with 4 populations who had been studied previously in the HapMap project. CEU are actually from Utah, but they sort of Northern European ancestry, so they are logically placed in Europe. A population from Africa, from Nigeria, Ibadan, and then ones from China and Japan. The phase 1 data that was published about a year ago included more populations from Asia, Europe, Africa, and populations from the Americas, which are interesting because, in particular, from the Americas, though this happens all across the world, of course, the ancestry isn't a clean ancestry. There's been a bringing together of peoples from different continents, and that leads to complexities in the ARG, in the genetic structure, in some sense, deep cycles in the graph, from your point of view. And the complete project, where the sequencing is now complete, involves 2,500 people, a few over that, 2,520, I think, aiming to get 500 from each major continental area, split into 5 groups from each continental area. So basically 25 sets of 100 individuals, more or less. And we've got some whole genome sequence from them all, and also deep sequence from the coding region, the exome. And of course, we get a wealth of data from this. When we started, there were about 10 million genetic variants known. The first pilot found 15 million, and it increased over 20 million, the total. Phase 1 was 38, found 38 million, and the final is like about 90 million product. And this is a small part of the genome, and there's all sorts of patterns of variation, and there are these long black lines, those are deletions. You can see that the African populations here in brown in the middle are a little darker. There's a little more variation there because there's more genetic variation within Africa. And there's all sorts of other relationships that we don't have time to go into, unfortunately. There's one sort of biological point I wanted to discuss a bit that I think might be interesting, and there might be some ideas here about improving this approach before going back to how we're going to handle this as we get more and more data. So, of course, we're interested in functional variants. A lot of mutation is neutral. It can mutate any base in the genome, whether it's functional or not. But we're particularly interested in mutations that might have led to a change that has a function. So here we're looking at the distribution. On the x-axis is allele frequency, so rare things at the left and common things at the right. And what you see is that there are more rare alleles. That's a well-known property of the coalescent and of what we expect in population genetics. But purple things are synonymous mutations. These are mutations which are in coding sequence, but they don't actually change the protein sequence. They're in the third base position, which is most of them are a position which doesn't actually affect the amino acid residue that's translated. And you can see that there actually are there's a reasonable number of common purple mutations. Some of you, the ones at the front, can see that. If you go up this list, the green ones are non-synonymous. Those change protein sequence. The blue ones are splice variants. Those will create incorrect splicing, basically destroying the protein function. The red ones are stop codons. Those also lead to a hard stop in translation, so they also tend to destroy the protein function. And the gold or yellow ones are things that have been previously annotated as being functional human gene disease mutations. And what you see is as you increase in functionality or damage to the protein, there's more and more tendency to suppress mutations that are common and to have a relative enrichment of rare ones. Now, of course, this is not unexpected. What's going on is that damaging mutations, they may happen, but they tend to get selected against. And so by natural selection, it's unlikely that they'll drift to high frequencies. There's basically a random walk, a sort of diffusion process happening to the mutation frequency, but there's a pressure downwards for damaging mutations. So this isn't really an enrichment of rare mutations. It's a suppression of common mutations for common functional mutations. And we can see that in the bottom half of the slide. This is all normalized by the synonymous rate. So you can see the synonymous rate is flat here. The greener, there's a bit of a bias for the non-synonymous ones, and progressively more bias as you go to the protein loss of function mutations and the known functional human mutations. So this is a dynamic process, though. It's not just that these were suppressed and that's it. There are always new mutations happening, and they're drifting in frequency. So to maintain this type of curve here, we need to dynamically have mutations which are damaging in this region, which are being suppressed. So because of that, we can actually estimate how many, on average, damaging mutations each person has. So nobody in this room is perfect. You all have damaging mutations you're carrying. Some of you, there's enough people here who there'll be some people who know they have genetic diseases. There's a few % of people, where there's a mutation which has a very strong effect. If your parents have it, you know that you have it. There'll be some people with genetic diseases. But many others may be carriers for genetic disease. They may have them in a heterozygous state, so there's no visible phenotype, but you might pass it on. Or you have some weaker mutation that's deleterious but has a weak effect. So we can estimate the load per individual. There are about 4 million genetic variants separating each pair of genomes, or pair of individuals anyway. And we can we can estimate how many deleterious functional mutations there are per person in different categories. And there's approximately the order of 1,000. And of those, a couple of hundred, a few hundred are probably in protein sequences. A few tens are probably loss of function, really killing the protein. And the rest, though, are probably in the non-coding sequence. And we don't know so much about that. There's interesting studies, there's an ENCODE project and so on. So we're beginning to work out where those are and what those are. So that's a kind of burden. We're all carrying a burden of deleterious mutations. So how do we actually identify? That's a sort of generic thing, though. You can statistically estimate the load, but you can't say which ones are which. Of course, we really want to know which are which. That's the study of medical genetics or functional genetics. So here again, we have this is a standard plot that some of you will have seen. Allele frequency on the x-axis on a log scale, so 1% is in the middle, common on the right-hand end, rare on the left-hand end, a fraction of a percent or below. And then effect size, how strong is the mutation? So people started studying mutations that have a strong effects like hemophilia and so on. And you can find those, and with the genome sequence and tools now, we can actually find the gene. And in the 90s, hundreds of genes, actually a few thousand genes have been found like that. But they're all very rare, it turns out. When we got to start understanding human genetic variation, we could design experiments that looked at common mutations, the first ones that we found, and we can go and use association studies like I told you. And we thought we might find lots more, but actually almost everything we find is very weak effect, has low effect. However, I just want to sort of say association studies have been important, and there's over 1 million people have been studied, over 1 million people genotyped. 23andMe, as a direct-to-consumer company, has genotyped 400,000 people. So there's hundreds of thousands of people. And there are thousands of genetic variants that have been identified as being involved in disease, and whether or not they contribute heavily to the population health burden, they connect biologically a gene with a function, and those are important studies. So to get back to our picture, there's nothing really in the top right-hand side that's deleterious anywhere, because that would have been selected away. There's not much in the bottom left that we can get at. I mean, maybe there is something there, but if it's very, very rare and it has almost no effect, you're not going to find enough of it to measure it. But there's a big hole in the middle, you might ask, and that's what the field has asked itself. And we actually know there's some things there. We know of genes where we've gone and looked because we had candidate ideas, and we found variants that had intermediate effects and so on. So there's a question, now we can sequence. Can we start to look directly for genetic variants which are at sort of 1%, fraction of a percent frequency? Some people call them Goldilocks variants, not too big and not too small, and really contribute substantially on this axis. So we were involved in such a study in the UK with 10,000 sequences. We sequenced 4,000 whole genomes, 6,000 exomes. We've collected all the data. Basically, it's there. We've got 50 million variants and so on. We started to look for functional variants. It was a little disappointing. That's a message which other people have been finding. We have found some additional new things. There are a few Goldilocks things, and there are a few things that were missed by the previous studies because they actually just weren't well represented or tagged or assayed before. But if we go through the maths, the numbers we have of this was on an earlier analysis on 2,500 samples. We've now done this on all 3,700. That number is just not enough. We know from the GWAS studies that you need to start looking at tens of thousands of people to see the effects that we're looking for. So what's an alternative strategy? Can we combine the two data sets we have? We're getting this data on thousands of people's genome sequences. The UK 10K is just one of a set of projects across the world, by the way, all with similar results. But can we combine the GWAS studies with our reference sequence, and so there is a way to do that, which is to impute full sequences into the GWAS individuals. It's basically a missing data problem. You've got some picture of the genetic structure built out of your reference sequences. You want to build some model, and then you want to get partial data from the GWAS, and what they do is they look at just 1 million positions that are known to be variable and assay those. So you're not getting a complete sequence. You've got a partial sequence of these individuals. So here's an example. Here's a set of reference sequences and a set of reference of individuals who've been GWASed, and we have hundreds of thousands of these. We have a few thousand of these. And what we do is we look for matches. We look for, in the substring that's in the genotyped individual, we look for a match at the relevant positions to somebody, this bit here matches that blue line there. So, and then once we found a match, in principle, we can copy down. And effectively, what we're actually doing is using a hidden Markov model to do this. So there's an inference framework for this. And it says that you're going to try and explain the new variants here in terms of a mosaic of pieces from our reference individuals. And we can do that in a probabilistic fashion. We don't actually know. It may be that it matches several of them, and it may be there's a longer match to some and a shorter match to others. We'd like longer matches if we want to. We have a process. The hidden state is which of these guys are you copying from, and you allow a switch. A transition is switching between those. And those transitions, in some sense, correspond to ancestral recombinations in our ARG model. So this is a method that's state-of-the-art. It's used in these programs in Pute and Mac. It's quite heavy. It's effectively an approximation to conditional sampling on the ancestral recombination graph. But there's no tree here. It's not actually a very good approximation in some sense, but it works. And it's been very important in the field. Here's an example of a place. We're looking at what's called a Manhattan plot. This is the p-value, -10 log 10 p-value on the axis. And so you want a spike. That would be a very rare, a low p-value. And the black is real data, and the gray is imputed, and we're getting a much stronger signal from an imputed variant, and it's likely to be the functional variant, even though it wasn't typed. And it's been also important for integrating data across projects and so on. So that's the way that things are done. Now, this all sounds great, wonderful, problem solved. The trouble is that imputation accuracy drops as allele frequency drops, just exactly in the region we're most interested, around 1%. If you take the 1,000 genomes data, it falls off a cliff. So we can impute all the common stuff, and that's actually useful, but it hasn't solved our problem. At least it hadn't done up until recently. Now, we did notice, as we sequenced more individuals, that it got better. One got a progressive improvement from, say, 70 to 82% in this category by going to more individuals. So the question is, can we as we go to much bigger sets, can we do better? Can we do better either by improving our model or by improving the numbers? We all know that both things are good. And we probably want to improve the speed as well because the methods are slow. So here's a pilot study that we've been in the middle of undertaking now. There's, I think, 9 studies, 13,500 samples. Here's UK 10K and 1,000 genomes, and a set of other projects in Holland and Sardinia and Finland and so on. And we've taken sites down to 0.03%, 1 in 3,000. And if we look at our imputation performance, it pushes us up. So now at 1%, we've gone up to 78% R-squared correlation between the truth and we can use holdout data and establish a truth metric. So this is looking promising. And in fact, simulations suggest we should even do better. And up here in this 80-90% level, that's where we want to be. We really could start not having to sequence, look at the Goldilocks region and imputing. We're on the verge of that this coming year, I think. And I wanted to say briefly, but I think I'm going to skip through this rapidly. Imputation is also used actually to integrate data, because in some sense it does have a working model of how all the data is, how to share data across individuals. And so what we can do is, instead of, if you want to sequence one person by themselves, independent of all other knowledge, you have to sequence it to very high redundancy so that you can statistically handle the uncertainty in all the sampling and so on. But in order to find low frequency variance, that's very inefficient. It's a waste of time in a way to sequence at 30 or 50x. And it turns out that you can share across samples using imputation, and that from a population basis, you're better off. This is a deep sequence of 30x on 50 samples, and this is 16x on 100 samples, or 8x on 200. Same amount of sequencing, you can push up your power to detect things as you spread data, as long as you integrate it carefully. And that's the way we did things for the 1,000 Genomes project, and we did meet our goal of over 95% at 1% frequency. And we get a lot of rare stuff as well. So all this more or less works. And we can also, as well as actually finding variants, we can improve using imputation the accuracy of calling the individual sequences. OK. So how to speed it up? So hidden Markov model-based methods, like computationally heavy, they're quadratic in principle in the number of reference sequences, and there's some things that they use to make them linear, but they're kind of heavy linear. They use MCMC sampling. They come out of a statistical sort of framework, and they take thousands of CPU days for the sorts of data set analyses that we do. We have big compute farms. We have many thousand machines and lots of, it's painful. And they're not going to scale to millions of genomes. We want millions of genomes. It's only coming. We have to do this in two years or so. So I want to discuss with you a new data structure that's related to other data structures used in computational biology, which I'm calling the positional Burrows-Wheeler transform. So let's imagine you could sort all your sequences. Let's say we're interested in some position. Of course, these sequences are very long. They're millions of positions long. But let's be interested in one position, K. And let's imagine we'd sorted them all at that position. Of course, once you've sorted them, then finding matches is cheap because matches are adjacent in a sort order. It's like looking somebody up in a phone book. And what we're showing here actually is the length of the match to the preceding sequence. And we've sorted from this position going leftwards, and this is the next position we're looking at. So this is analogous, for those who know about it, to the Burrows-Wheeler transform on strings. But it's different because here we have a large number of sequences, all of which are aligned. So we have a kind of a coordinate system, which is different from a standard Burrows-Wheeler transform of a string database. OK. So one thing you notice is that there's some clustering here. Because we've sorted them, and we know that things share sequences, that means when we look at the next position, there's a tendency, these are all sharing material, there's a tendency for the next position to be shared as well. So there's this block of ones and a block of zeros here. And actually, this is a small data set. When you get a big data set, it's extremely blocky. I forgot to mention. So what about this sorting? Sorting seems nice, but it's expensive. How do you sort at every position? Well, you can update your sort. It's kind of obvious, really. It's a computationally simple thing to do. If you're sorted at some position and you want to ask what's the sort of the next position, you just have to look at the next character. All the things that are 0 go to the top, and all the things that go 1 goes to the bottom. But you maintain the sort order going backwards from that, because you're just adding one symbol on. So that's a very fast operation. It's done in a single pass down the sequence. So single pass through the entire data set generates all the sort orders at all positions. So we can use this for compression. And compression is going to become important later on. Compression is always important. It's good to have compact data structures, and it's also indicative of prediction. So we use the run length encoding on these columns. We're storing the data basically column by column in this, each one relative to its current sort. We use run length encoding of an extremely naive sort. I'm sure there's many. I coded it myself. So we simulated 100,000 sequences with a standard coalescent simulator. There are 370,000 sites, so we've got 10 billion data points or something, 37 gigabytes of raw output. So gzip, there's lots of ones and zeros. Gzip compresses it 35-fold to a gig. You might think that's quite good. But once you use this run length encoding, sort of naive run length encoding, it goes down to 7.7 megabytes. That's a 4,800-fold compression. So, and actually, that naive, if I took the native order and run length encoded it, it would be 2 gigabytes. So that's worse than gzip. So not surprising. So that performance just gets better the more sequences you get. We can subsample our 100,000. You can go up here, and the compression ratio increases from 6-fold over gzip here at this end to 130-fold at this end. So that's what we want. It's all behaving nicely. And this works with real data. We get a factor of 6 on the 1,000 genome set of 2,000 samples, which is more or less where we'd expect it to be based on the simulation. So what about matches? Well, maximal matches, as we said, let's take a new sequence and ask for matches. Maximal matches will be adjacent in this structure. What we want to do is we have this structure. We want to place a new sequence into this structure. It's going to basically follow along, and it'll map up and down. And it'll sit next to it. Each place it'll sit next to the thing which it matches best currently in the process. And there's also a simple linear update rule for that. It's the same rule actually as we did for the standard sorting. So we can take two approaches. We can build an index. We can store all the relevant information for the update. It's quite a big array. It's probably, you can be more efficient, but it's certainly the size of the original data structure. Or we can just match dynamically by going through the structure, match a set of things. So here's the time to match 1,000 new sequences. So if you take a naive approach, that'll increase linearly with the size of the reference set. The reference set here is going from 1,000 to 50,000. It took 50 seconds on my laptop to match the chunk of sequence from the simulation. And it went up to, 10,000 was 500 seconds. And it's exactly linear, as you might expect in a naive implementation. So in the indexed implementation, that's constant. It's basically taking a user second independent of size. And that's not surprising. I mean, it's like a hash matching or a Burrows-Wheeler matching, an FM index matching. It's like a text matching type algorithm. But we're doing it on these very long strings into this data structure. And we're finding all the maximal substring matches for 1,000 sequences in about a second. It increased at the end because I started swapping. I ran out of memory for my data structure. The batch process didn't have any, just will scale as far as you want, but does increase at a slow rate, but still is 12 seconds versus 2,500 for the naive approach. So this is a really fast approach. Okay, so what about inference? I mean, this is all sounds sort of algorithmic, but this is an inference machine learning meeting. Well, prediction is related to compression, as I said. So let's think about this next site position here. Let's call that y, with yi being the ith element. And we're going to have di being how much you share to the previous string in the sort order. And we can consider a generative model for y given d. So that's quite a simple thing. We've just got a one-dimensional vector of ones and zeros, and we've got some other information. So a very simple generative model, which actually is the only one we've been playing with so far, is a Markov model, where you say the probability of yi plus 1 is conditional, or yi plus 1 is just conditional on the previous element and di, yi and di. So actually, it's kind of interesting to think about this. You can use this to generate entire sets of sequences. You could, at some point, you can generate y, and then after you've done that, you can update your data structure, and then you can generate another y. So you'll generate a family of sequences. This is entirely different from a coalescent generator, or from a hidden Markov model generator, which would be some sort of random field process for iterating, which people have, but extremely slow to resample. Okay. So, can we do anything with this? So it turns out that we empirically sort of started to look at what the probability of a switch is. Essentially what matters is switching from ones to zeros. So p that yi plus 1 isn't equal to yi given d appears to be pretty cleanly exponential in d. As d gets large, that means that this gets smaller, which is good. As you get longer matches, then the chance of switching drops. The chance of extending the match increases. And actually, that's expected given d. This is, in fact, what you'd expect, because this is sort of the derivative of the length distribution in some sense. It's the chance of extending a length. So, and that would correspond to an extreme value distribution on, which is what you'd expect for best matches. So this all fits. So, so we've implemented an extremely simple imputation process. Another point is you can think of going from left and right and combining data, because this is sort of bidirectional, this structure, so you can impute from both directions. And I've run it on this on the real data set on 13,000 samples. There's the results that Jonathan got with Impute, which is this heavily invested software. That took days to phase and run on that data set. This took 25 minutes on this laptop. It's slightly worse, to my annoyance at the moment. But I think it's definitely in the same space. It's definitely better than the original one. It's a little bit, it kind of comes down here, just underneath the yellow line. And there's 26,000 haplotypes and 500,000 sites involved in that. Okay, so what could we do better? Well, I think there's quite a lot of tricks and elements that are possible. But one interesting thing is to look at the other models on this vector y. And a very interesting natural one is to think about trees on it. Because that would correspond to a tree. There's nothing, we've sort of got away from a tree. We know that there should be one. Can we have a tree sort of prior structure? And the Ds are informative. They might tell you, when D is large, they might tell you that things are closely related. And when D is small, it might be that it's a deep split. So this is an open thing. It's just an idea. I don't know about fast ways to explore tree priors. I mean, there's a lot of work on that. The data aren't consistent with the tree in the form, at least I've drawn it. So you have to do something more sophisticated. But this is the right audience to think about those things. So I've got a few minutes left. 5. So I want to go back, relate this to some other work and ideas. Going back to raw data. So far I've been imagining I actually have sequence or partial sequence for these individuals. Certainly the reference, I'm imagining I have these lined up perfect sequences. But actually the raw data we have are shotgun sequencing reads. And these are short. The way that we sequence a genome is we don't start at one end and go along. We get 100 base reads, and we have to kind of work out how they all fit together and onto the reference. They're random fragments. They can be enriched if you want. And what we do is we map them to a reference genome. We say, we think this read looks like this piece of the reference genome. But there are problems with that. There can be problems where the genome is repetitive, so the same sequence is found in several places. We're not quite sure which one the read goes to. And there can be problems because the read actually doesn't come from the genome. It comes from a different version, and that's what we care about. So we're actually interested in the things that don't match the reference. So matching to the reference is a slightly strange thing to do. And then we detect variants based on multiple alignment of reads, and there's methods for that. So a lot of modern mappers use the Burrows-Wheeler transform. And I've got a couple of slides about this, but I think for time I'm not going to go through them. Basically, it's based on the suffix array, and you can update with a formula that looks actually extremely like the one, not coincidentally, that we used for updating our sort order. It's it's essentially the same formula. And it's a constant time update, and you can update the matching of a read. And so BWA, which is I think the most widely used program developed by Hung Lee when he was with me, and Bowtie and other programs all use this. But you get errors due to mapping problems. These errors picked up because we found things present in Britons but not in CEU, which should be the same. And they all, in fact, turn out to be around the centromeres and the telomeres, ends, the hard bits of the genome that are repetitive. You get errors because of alignment. This is a region where there would be lots of variants called. These are reads aligned to the reference. But actually, what's happening is that there's a duplication. And there's a whole set, all that sort of chatter of apparent genetic variation is due to a single clean insertion, but the alignment was problematical. So a better way to work is not really to use the reference. It just creates trouble, is to think of your read set as representing your individual. You'd like to work with the read set as a representation. And think about assembly, work in a reference-free fashion. And there are ways to do that. You can start from some read which is unique in a sample, and you can kind of build out from it and find the alternate version that's in the sample. And here we've picked out a variant that explains this bubble in the assembly. And the nice thing about that is this is 2 yeast strains. You get a single base change, looks like a sort of divergence in the assembly like this, but also a big insertion, which would be very hard to map to. You couldn't map a read onto that because there's nothing there to map to. Looks exactly the same in the assembly version. The assembly version is neutral to the nature of the variation, and it's attractive. And if we do this, it does find things and clean up problems that we know that we have in assembly. So it turns out that some of the state-of-the-art assembly methods also use this Burrows-Wheeler construction, not applied to the reference sequence, not to map to, but they take all the raw data and they build essentially an alignment, a Burrows-Wheeler transform of the read set. And that's related closely to an assembly graph, and you can compute from it in order n time in a single pass effectively, or the non-transitive overlap structure of all the reads. So you can get a minimal overlap structure which identifies all the components of the assembly. And you can also derive all the components of standard assembly graphs, de Broglie graphs for length k. So there's a paper by Jared Simpson, who's now in Toronto, about that. There's some open questions in that field about whether the graph is the same. But the question which I kind of think is important and interesting is, what are we going to do as looking forwards as we get these large amounts of genetic data? We're going to want to have a more sophisticated reference to map our reads to. We're sort of going to want, and can we merge the positional Burrows-Wheeler, the PBWT ideas I introduced earlier, which are about shared genetic structure over long distances. So that was taking advantage of recent common ancestry. Can we sort of combine that with working just with the primary data rather than getting ourselves confused by mapping to the wrong reference? So that's a challenge for this field. It's a very big data problem. I think it's ultimately a clean way for us to work. And it also would mean that all the experimental data sets, Terry mentioned RNA-seq data sets, you would interpret them in this context. And my expectation is you'd need rather little data per individual to get a very good fit into the model and to start making inference about individuals. So it would become extremely efficient as we build these structures. So you'd have large distributed data sets. The more people, the more information. The less information you need per person, the better sharing. But we have to be smart about it. It has to be fast, and we have to have algorithms at scale. And it seems, this is the sort of genome-wide version of what I imagine goes on with page-type search for Google and all these other web searching systems. And I think it's going to become a big thing. It's already a multi-billion-dollar enterprise. It's going to be a very large-scale effort, and I think to get the moment, I would say, there isn't real proper computational engagement. There's smart statistical engagement, and people, manage to put together what's needed to get at things, but there's a lot of scope for using some of the technology that this room has available to it, and the thought processes. So is anyone interested to help? That's a question. Collaborators, postdocs, independent or in collaboration would be fine. Of course, there are lots of other big data machine learning problems in genomics, but I think this is a very core thing. The basic nature of the genetic structure of mankind is a core thing that we should work out how to represent and work with well. So I'm going to stop there. These are people in my group. Jared and Heng have left. These people have done a lot of work on these large data sets. There's big consortia involved. This is the 1,000 genomes. It was a long time we had more people than genomes, but that's no longer true. And this is the UK 10K group. So thanks very much.
All right, thanks, Richard, for a wonderful talk. There is time for some questions. Terry, I'll give you my microphone.
So this is really exciting. It's a tribute to exponential growth of computing power. Mm-hmm. But let's ask, what will happen as n goes to infinity here? In other words, suppose we achieve, this compilation of all human genomes. Mm-hmm. What can we use that for, and what do you think will emerge from that era, which is probably not that far off?
Well, I have another talk, actually. So people think of genetic variants as happening kind of here or there by random. But with 7 billion people, we actually know that there's about 100 copies of every possible change at every base in the world, as long as they weren't lethal. And you might say we're a long way off sequencing everybody, but actually if you sequence 1 million people in a smart way, you can get towards saturation. So one thing we'll do is there are people with variants in every gene. And so from a biology point of view, we can exploit that. We can try and help find out function of every base in the genome. We can, from a clinical point of view, people who have severe effect mutations, we really want to match those. This happens already. We want to find the other people in the world who carry that, who put them in touch. They can learn from each other, things that have worked well. Therapies that have actually worked, we can transfer, and even if they haven't, prognosis is an important thing. So it's extremely important diagnostically to identify functional variants and to match. And so having structures that allow us to match efficiently is very important, actually.
So what about positive selection? You talked about negative.
Well, positive selection has that affects the structure of the graph as well. And you can look for signs of it. And there are examples of positive selection in humans. And yeah.
All right, over here. So
where are you?
Over here, straight in front of you.
Okay.
So using the current state-of-the-art imputation algorithms, could you estimate roughly how many full sequences we would need in order to be able to do a good job of imputating everyone in the world?
Well, it's all these trade-offs. As you get the best data point on that is it depends on population structure. In Iceland, the population of 300,000, they've sequenced 6,000 of them. They've got genetic data on 120,000 or so. And they can impute those 120,000 extremely well. But because you can basically have a second cousin who has been sequenced, or a third cousin. So it depends. There's it's a sliding scale, essentially. I'm not going to, in Europe and in America is the worst place in the world, sadly. You have the most the biggest genetic pool that's mixing most readily. But you will get the order of 1 million people is very strong for anything that's not recent.
Okay, one last question, please.
You have shown very impressively these compression of these genomes. Did you think about the best possible compression? So what's the minimum, like a lower bound what you need in order to compress these genomes? So you have generated that.
I don't know. That's an open question. If you take the standard model, coalescing with recombination, I don't really understand how that model, the coalescing with recombination, which is a distribution, how that correlates to my Y distribution. I don't have a good model. There's a very strong mathematician who's joined my group recently who wants to think about that.
Yeah, that's interesting. Okay, thanks.
All right, the next talk is called BIG and QUIC, Sparse Inverse Covariance Estimation for 1 million variables. The authors are Akshay, Sustik, Dylan, Ravikumar, and Poldrak. And Shay will be giving the talk.
Okay. Hi everybody, my name is Choi Xie, and today I'm going to present our new algorithm Big and Quick, which can solve the estimator inverse covariance matrix for 1 million random variables. And this is a joint work with Matthias, Indra, Pradeep, and Russell. So there are many applications for the inverse covariance estimation problem. I'll first give an example in the fMRI brain analysis. So in this example, we are given some fMRI images. And our goal is to reveal the functional connections between different pixels in the brain image. So we output a graph like this. And we can use the inverse estimation technique to do this. So first we consider in the input image, each pixel is a random variable. So if we have p pixels in the input image, then we'll have p random variables. And we can estimate the inverse covariance matrix, which is a p by p sparse matrix. And this matrix corresponds to the adjacency matrix of the output graphical model. So there are many other applications for inverse covariance estimation, including the gene network discovery and many other applications. So now we formally define our problem. Assume we have p random variables, and those random variables follow a multivariate Gaussian distribution. So we observe a n samples from this distribution. So we observe y1 to yn from this p-dimensional distribution, and our goal is to recover this distribution using the samples. So we are particularly interested in the inverse covariance matrix because the following reason. So for example, if the underlying graph is the chain graph, then the inverse covariance matrix will be a tridiagonal matrix like this. So we can easily see that the non-zero elements in this inverse covariance matrix corresponds to the edge in the underlying graphical model. So we want to recover the inverse covariance matrix using the given samples. And we are considering a high dimensional setting where the number of random variables is much larger than the number of samples. And since the non-zero elements correspond to the edge in the graphical model, so we want to select an inverse covariance matrix which is very sparse. So people usually use this L1 regularized maximal likelihood estimator to estimate the inverse covariance matrix. And in this estimator, we want to solve the optimization problem like this, where we want to find a p by p matrix X, and this X will minimize the negative log likelihood. And in the same time, we want X to be sparse, so we add the L1 penalty into the objective function. So if we can solve this problem, we can estimate the inverse covariance matrix. But this problem is very hard to solve because the following reasons. First, we have the L1 penalty here, which is non-smooth. And moreover, number of parameters in this optimization problem scale quadratically with number of variables. So if we have p random variables, then this X matrix will be a p by p matrix. So we have p squared parameters in this optimization problem. So there are many algorithms proposed to solve this problem. So 5 years ago, people can only solve problems with about several hundred of random variables. But in these recent 2-3 years, people proposed a lot of new algorithms, and now we can solve a problem with p equal to 1,000 or even p equal to 10,000. However, if we want to even solve a larger problem, we face a memory problem. So all the current solvers require to store a p by p matrix, so they need order p squared memory. So this is why all the current solvers cannot handle problem with p larger than 30,000, because we cannot store a p by p matrix into memory. But in many applications, we want to solve this problem for very large number of random variables. For example, in the FNRI dataset, each input image have about 200,000 pixels. So we want to solve a problem with 200,000 random variables. So motivated by this application, we propose a new algorithm, BigQUIC, which can solve a 1 million dimensional problem in 1 day using a single machine with 32 gigabytes memory. So our new algorithm, BigQUIC, is an extension of the QUIC algorithm we proposed 2 years ago. So in this talk, I'll first briefly talk about the QUIC algorithm. I'll show the difficulty to scale to p equal to 1 million, and I'll show how to handle those difficulties. So the QUIC algorithm is a proximal Newton method, where we first split the smooth and non-smooth part in the objective function. So we say g of x is the smooth part, and h of x is the non-smooth part, which is the L1 penalty in our case. So in each iteration, we form a quadratic approximation for the smooth part, and we define the generalized Newton direction to be the minimizer of the quadratic approximation for smooth part plus the non-smooth part. So in this talk, I'll use xt to denote the current solution, which is the p by p sparse matrix, and d is the Newton direction. Unfortunately, we cannot solve this subproblem in a closed form. So in a QUIC algorithm, we propose to use a coordinate descent update to solve this problem. So in a coordinate descent algorithm, each time we only update one variable in the matrix. So if we only want to update dij, then we have a closed form solution using self-thresholding operator. And in this closed form solution, the most difficult term to compute is this one, Wi transpose dwj. And this W is the inverse of the current solution. So even if the current solution is sparse, the inverse of it will be a P by P dense matrix. So in the QUIC algorithm, we show that if we can store this W matrix in the memory, then we can do this update very efficiently. And this is the summary of the QUIC algorithm. We first select a subset, which is we guess the most element will be 0, and only a few elements will change from 0 to non-zero in this iteration. And then we run a coordinate descent algorithm to find the Newton direction on this subset of 3 variables. And then we do a line search to find the step size alpha, which satisfies so that the new solution x plus alpha d will be positive definite and will satisfy a sufficient decrease condition. To check those two conditions, we need to do a corrective factorization on the P by P matrix. So now we analyze the time and space complexity for this algorithm. So we have 2 parts. In the coordinate descent part, we have to store the W matrix, so we need order P square storage. But it's very efficient. We only need order MP computation per sweep. M is the number of non-zero in the current solution. In the line search part, we need order P square storage and P order P cube computation. So now we want to handle the problem with P about 1 million. And non-zero is the constant times P. So we cannot allow the P square storage, and we cannot allow P cube computation. So we first deal with the P square storage problem in the coordinate descent update. In the coordinate descent update, now we don't have enough memory to store the P by P W matrix, and we assume we can only store the M columns of W into memory. When we want to update the ij element, we want to compute this term Wi transpose dwj, and this can be done efficiently if Wi and Wj are in the memory. Otherwise, we have to recompute Wi and Wj by solving this linear system using conjugate gradient method. So this is an example. If we can store 4 columns of W into memory, and we store W1 to W4 in the memory, and the green elements are the free sets which we want to do the coordinate descent update. If we want to update the 1,4 element, and luckily we found W1 and W4 are already in the memory, so we call this a cache hit, and we can directly compute the update without recomputing Wi and Wj. So this is very good and very efficient. However, if we want to update the 6, 9 elements in the matrix, we found W6 and W9 are not in the memory. So we have to recompute them using conjugate gradient method. We call this a cache miss, and this is very bad. It's very time consuming. So ideally, we want to find an update sequence in the coordinate descent method, which can minimize the number of cache miss. However, this is a combinatorial scheduling problem, so it's probably NP hard. So we propose the heuristic update sequence, which is update the variables block by block. So our algorithm is like a block coordinate descent algorithm. Each time we update variables within a block. So for example, if in the ideal case that the free set can be written as the block diagonal matrix, then we can first compute W1 to W3 and update the first block, and then compute W4 to W6 and update the second block, and so on and so forth. So in this block coordinate descent, we only need totally P column computations. Each column only need to be computed once. But in the real applications, it's usually not this ideal case. So we have some elements outside the diagonal blocks. And our main finding is that the extra column computations can be characterized by the notion of boundary nodes. So I'll not show the formal definition of boundary nodes here, but intuitively, if we have 6 nodes, 1, 2, 3, and 4, 5, 6, and we found 2 is connected to 4 and 5. Then we say these 3 intermediate nodes are the boundary nodes. So we can write down the number of current computations to be p plus total number of boundary nodes, and we can minimize total number of boundary nodes by using a graph clustering algorithm. And this is very important in practice because we can significantly reduce the number of current computation. For example, in the FNRI dataset, we have 0.228 million random variables. If we use a random partition and run block coordinate descent, then we need 1.6 million current computations. But if we do a graph clustering to minimize the boundary nodes, and then run block coordinate descent, then we only need 0.237 million current computations. And this number is very close to p, means we don't have many additional current computation to do. So we can, under a fixed size of memory, we can have similar time complexity in the coordinate descent update. And then in the line search procedure, we want to compute the log determinant of a p by p sparse matrix efficiently. So here we propose an algorithm to approximately compute the log determinant of a large sparse matrix, and our time complexity is only order mp. M is number of non-zero, and we don't need any additional storage. So I will not show the detail here, but this approximate computation for log determinant is very interesting and may be applied to many other applications. So we also reduce the time and space complexity for a line search. And this is a summary of our algorithm. We also do a variable selection, and we construct a partition by clustering. We run a block coordinate descent algorithm to find a generalized Newton direction, and we have an efficient line search procedure. And in the paper, we also have some theoretical analysis, because in this large-scale problem, we have to compute the hashing using iterative methods, like conjugate gradient method. So we show that even if we don't solve the linear system exactly, we can, by carefully controlling the residual in the conjugate gradient method, we can, like this, we can achieve the quadratic convergence rate in practice, even if we don't exactly compute the hashing matrix. And this is the scalability results. So we increase the size of number of random variables from 1,000 to 1 million. We can see that the QUIC algorithm will take more than 3 days if the p is equal to 30,000 here. And if we use a BigQUIC algorithm, we can solve a 100,000 dimensional problem in 10 hours. But if you use a more powerful machine with 32 cores, then we can solve a 1 million dimensional problem in 1 day using just a single machine. And we also test our algorithm in the median size dataset, where p is equal to 20,000. And we see the BigQUIC algorithm, even if 1 core, is still faster than QUIC algorithm. So this gap is because of mainly because of we have a more efficient log determinant computation in the line search step. Finally, I'll show the experiments on the FNRI brain analysis problem. So in this problem, we have each image have 200,000 voxels. And we try our algorithm with different regularization parameters to construct the inverse covariance matrix with different sparsity. And we discussed with the neuroscientist Russell, who is also a co-author of this paper, and we have the following 2 findings. First, we find that in the inverse covariance matrix, the voxels with the high degree nodes, the red and yellow nodes, are generally found in the gray matter. And people usually think the gray matter is more important in the brain, in our brain. So it makes sense that they have higher degree in the inverse covariance matrix. So we further do modularity clustering using our inverse covariance matrix, and we can find some meaningful clusters corresponds to some brain modules. For example, in this upper panel clusters, this looks like the same motor sensor network in the brain analysis. And the lower, and this cluster looks like a combination of three meaningful brain modules in neuroscientists. So this is the first time we can run the inverse covariance estimation for this large size fMRI data set. So in the future, we expect there are many more interesting results generated by our algorithm. So in summary, we propose a BigQUIC algorithm which can solve a 1 million dimensional problem in 1 day. And our innovation including the block colonial descent with clustering and efficient computation for log-determinant and inexact hashing computation with quadratic convergence rate. Here are some references for this talk. Thank you very much, and I'm happy to take any question.